Okay, so good afternoon, and I welcome all of you to this departmental seminar in the CBMS department. And also, I wish all of you a belated Happy New Year. And it's a pleasure to have as the speaker today Professor Ruchi Anand from IIT Bombay. So let me introduce briefly Professor Anand. So Professor Ruchi Anand did her PhD in 2004 from Department of Chemistry and Chemical Biology at Cornell University, USA. And then she did her postdoctoral research at Sloan Kettering and University of Pennsylvania. And then she joined IIT Bombay as an assistant professor in 2008. Her group employs a combination of X-ray crystallography, cryo-EM, and various other biochemical and biophysical tools to understand molecular mechanism with implications to human health. And her group has made significant contributions to two major areas, namely antimicrobial resistance and biosensors development. And regarding this drug resistance problem, she focuses on finding out new enzyme systems that can serve as new therapeutic targets. And also, she's involved in understanding the origin of antibiotic resistance. And her recent work is involved in working with bacterial transcription factors that has paved the way to structure guided development of biosensors, particularly for aromatic pollutants. And her work has been recognized with several awards and recognition. I'll just name a few. Since 2016, she has been serving on the editorial advisory board for ACS Sensors. She has won the National Women Bioscientist Award from Department of Biotechnology in 2018 and CRSI Bronze Medal in 2020. She's also the recipient of DBT Wellcome Trust Alliance Senior Fellowship in 2020. And she has been elected as Fellow of National Academy of Sciences in 2019 and also Indian Academy of Sciences this year in 2022. So with that, I welcome Professor Ruchi Anand again, once again, for agreeing to deliver this departmental seminar. And yeah, the floor is all yours, Ruchi. Please go ahead.
Thank you, Shuman. I'll share my screen and thank you for the introduction.
Yeah, you can.
I guess I'll, can everybody?
Yeah, you can go to your slides.
Yeah.
Yeah, and the title of our presentation is Allostery in Biological Systems.
All right.
So it's all yours.
Yeah, thank you. Thank you, Shuman. And without further ado, I will just start my talk, okay? And thank you a lot for the introduction. So today I thought that I'm going to, we do a lot of structure-based work and we combine structure with a lot of other techniques like biophysical, biochemical studies, as well as we do a lot of MD simulation in collaboration with Jagannath Mandal and at TIFR Hyderabad. And both the projects that I'm going to be talking about today has his contribution to make it viable or a successful story, as I'm going to be telling you about. So I just want to acknowledge this in the beginning of the talk. So like Shuman already mentioned, we work on various projects, and but something that I want to tell you is that in biological systems, allostery is very, very actually common. So there are many biological systems that we study where allostery becomes an integral part of understanding the system. I'm not going to be talking about my first part where we look at origins of antibiotic resistance and we try to understand how pathogenic methyl transferases, they methylate ribosome and result in many antibiotics like erythromycin, etc., which were working to stop working, and then they become superbugs. So we are looking in these mechanisms, and here also we see allostery plays an important role, apart from trying to understand the biological significance. We see underlying allosteric mechanisms that are there. Again, in our work on novel pathways which can be perhaps used for drug design, because they are mostly nucleobase pathways, dependent pathways for shuffling of nucleobases, which are important for DNA RNA synthesis. So these pathways are important, and here I'm going to take up an example today and spend quite a lot of time trying to impress on the allosteric mechanism in this system. And at the end of my talk, I'm going to talk about allosteric mechanism in the biosensor field that we are working a lot. Mostly, we are trying to do applied work, but there is a very interesting underlying allosteric mechanism that governs this system, and I'm going to talk briefly about it at the end of my talk. All right. So let me start with the first part of my talk, which is basically what we're trying to understand is we're trying to look at allosteric mechanism, in these essential enzymes which are in the purine biosynthetic pathway. And the first question that the first observation that was there in the system was that it's a bifunctional enzyme and there are molecular tunnel. There is a presence of a molecular tunnel in the system. So one would wonder, why do an enzyme would have a molecular tunnel? And I'm showing you a generic example, which is called carbamoyl phosphate synthetase. I say generic because it is one of the first ones that was discovered, and based on this particular enzyme, several other enzymes were then discovered and their functions were discovered. And what was found here was that this one has a 96 Armstrong tunnel, which is pretty big, and it goes from one, in this case, ammonia is produced, and then indole is transferred in the second part. So a huge internal tunnel is created for like highway conduits to be able to pass substrates. So why would that happen? Why would there would not be two enzymes? These can be broken into different enzymes, and why would there would not be just a normal small enzyme which could be functioning? And why would you want to have these conduits within the enzyme? So there are many reasons that people come up with, and some of them are that you have substrates which are so sparse that if you would let it go into the media, it may just get lost. So this is like you can say, kind of molecular. So it is good. It could also be that you have a generation of a substrate which is reactive. And if you let it go to the solvent, it may not have its right chemical form. Or it could just be that it's a difficult reaction and you want to flood the system with your substrate. So there can be many reasons why the product could be toxic to the environment also. So there could be many reasons why internal tunnels can be formed. So in our system, what we are looking at is this purine biosynthetic pathway, which forms precursors to bases. This is inosine monophosphate. It's a 10-step pathway, and ultimately, from inosine you can get conversions to other purine bases, and those can be then integrated into the DNA or the RNA. So you can have other manipulations that can happen before, but first this indole ring has to be formed, and here is a 5-membered ring in the first 5 steps, and the remaining 6-membered ring is formed in the second step, the second 5 steps. So here, this is where we are going to be looking at, and the enzyme that we are looking at, whose mechanism we are trying to understand, is this particular one, which is formyl glycamide synthetase, and it is the reaction it does is 2 of them. In the first active site, it converts glutamine to glutamate and releases ammonia. This ammonia then travels within the enzyme all the way to the second active site, where the C double bond O, in an ATP dependent, ATP is used to actually activate the carbonyl bond, and then in a nucleophilic form. So what I mean is you need the nucleophilic character of ammonia. Ammonium does not work here. Okay, so you cannot just let ammonia escape to the environment, because it will become ammonium, and that is not an effective substrate. So you have ammonia, and then this results in incorporation of the nitrogen at this position, which is required for extension of the ring later, and then you can have the 5-membered ring formed. So basically, what we were trying to understand is how these two reactions, reaction 1 and reaction 2, occur, and how are they connected to one another. So A couple of years, I would say, not couple, now close to 2 decades, but not quite there. We solved the structure of this enzyme while I was a graduate student at Cornell, and I've not been able to give up the system since then. So it has this green domain, as is shown here, where ammonia is released, glutamine. And then somehow, when this ammonia travels to this binding site, where it is converted into FCAM. And there is a domain here, the yellow domain that you see. It's a connector domain. Okay, and there's another site which is called the ADP binding site. So there is a gene duplication, but nobody knows what this site does. And even today, we have not been able to answer that question. And I'm not be answering this question, maybe in a future work. But the bottom line that I want to say is it's a very big enzyme. It has 2 active sites. Bifunctional enzymes are generally 55 kilodaltons or so, but it has these additional domains, which nobody knows. Some of the things we know now, some we don't know why they exist. So the surprising thing about this enzyme is, apart from its large size, it is also, when we solve the structure, the N in the C terminus and most of these long loops that you see here were all ordered in these enzymes. So it was like a very tense state in which we had trapped the enzyme. And also we had captured the enzyme, the glutamine. It goes via thioester intermediate. There's a cysteine, histidine, glutamate catalytic triad, that as it is called, which helps in the hydrolysis of the glutamine to glutamate, and in this case ammonia is released. And it was thought that this reaction, after this intermediate is difficult to trap, but we were able to get this in the enzyme. But no apparent tunnel or what are the connections between the 2 sites we could discern. Not only that, the only disordered element in the whole structure was a 22 amino acid loop, which is right above the active site, which is this roughly kind of stated over here. So Nandini, when she picked up the project, that was this structure was solved in 2004, and this is now more than a decade later when Nandini picked up this project, and the project had hit a standstill. The reason was people were not able to understand how this 2 enzymes, what is the coupling, and what is the path which kind of connects the 2 sites, whereas how the ammonia is getting transferred. They were not able to trap any of the intermediates, and no headway could be made because of its labile nature. Anytime we tried to put in any substrates here, we could never get in crystals or could not trap it in many states. So Nandini said, okay, let me try to look at this 22 amino acid loop, which is disordered. And let's do a little bit of a bioinformatics analysis using about more than 1,000 Purell sequences. She realized that there are a lot of glycines in this region which are highly conserved. By the height, you can tell. And then there are regions which are not so conserved. So in short, there is you can divide this loop in an imaginary loop because it's disordered right now. The conformation here is into regions which are conserved. There are 2 conserved regions, and then there is a non-conserved region. So what Nandini decided was she started making various deletions, mutations in this region, and see what's the effect on the activity of the protein. Does it become completely dead? What happens? So what Nandini did was Nandini took portions of this loop, and here I have given in black the region that we modeled, and we thought that was not in contact of the active site, rather towards this end terminal, as we call it, the adapter domain between the glutaminase and the active site domain. And then this red and yellow region was facing the active site in the modeling, and this was just extra region which was not contacting the active site. So what Nandini did was, with her hypothesis and the homology modeling that was done of the loop, we came up with this. And what was apparent is, if you see that where we think most likely the ligand is binding, that region, those red and yellow ones, the minute you start mutating them, you can see here, ammonia, this is the top graph is showing ammonia production. Nothing much happens to ammonia production. The gray one is how much ammonia is being produced or how much glutamine is getting hydrolyzed or glutamate is getting produced. That is what is being shown over here. And these mutations have no effect on this activity. Well, it is also 25 plus angstroms away. But when we start making mutations in this region, you see progressively ammonia production is stopping. So if you look at the first ones, ammonia production is this red and this golden star. Ammonia production is not a problem, but because your substrate is not able to bind FCAM synthetase, this downstream activity. So activity here, no activity here. But when you start making these black mutants, then you see progressively as you make these black mutants, which are facing away from the active side, not only ammonia production, which is 30 angstroms away, is getting affected. You also have, obviously, if no ammonia is produced, your FCAM synthesis activity is getting affected. You can see it's progressively getting affected. And this blue region, it is an extra region, it looks like. Not much to do with the activity. So then what Nandini did was, Nandini tried to understand why would activity so far away from the binding site go as you progressively make these black mutations. So what was observed was something was happening to the enzyme such that glutamine binding itself was going away. So you can see here, progressively glutamine binding is becoming worse and worse. And by the time you make all the 3 mutations, glutamine binding is completely gone. And activity as a result is completely also gone. So we thought that this looks like a slow catalyzing mutant. Something has gone wrong with somewhere in the communication network that glutamine can no longer bind. Or maybe something happened to the site. I don't know, right? So what I should do is, let me try to see in this slow catalyzing mutant, can we crystallize it? And we were able to crystallize this mutant, and we were able to get the loop, that the 22 amino acid loop, which was always disordered, to become ordered. And we could clearly see density for it, and we could see even glutamine in one binding site, and therefore this FCAR on the other side. So what that told us was, this is just showing the density, that this loop is like capping the active site. And remember, regions that we said were facing it, were really facing it and were involved in actually anchoring the substrate. So it kind of made sense. But yeah, that is good that we had this information. At this point, we involved Jagannath, and we said, can you, now that we have the closed form of the loop, let's do a simulation and see if we can capture states of this enzyme and maybe find the channel, maybe find the allosteric network. And what Jagannath did was, he did multiple 1 microsecond simulations. And what we observed here is, let me go back. This blue thing is our loop. Over here, what we observed here is that as the simulation proceeds, after initial stabilization, we see that this loop starts opening up. Okay, and there is a difference in the APO versus the glutamine. There are connections that are made in the glutamine bound which are not present in the APO1. But to make a long story short, we do see that a lot of motions happen in this N-terminal domain, and these linker regions and the loop region that I was talking about. So let me show you this movie where it shows as the loop opens, it starts hitting this N-terminal domain, which shifts and then crosstalk to the glutaminase domain develops. So if you look at this is the interface. While the loop is moving, there is a dynamic interface which is made. So this is the interface before the loop is moved, and this is while the motion is going on, and we see that an interface is developed. And what we thought was, last time, the residues that we were trying to mutate, the black stars that I talked about, were right in this region. So let me show you the black stars that I talked about. This is where we mutated, and we saw ammonia production has gone so far away. It's just gone. Okay. And here is the red mutation that I'm trying to show. So instead of making mutations in the loop, we said wherever this contacts this region, okay, of the N-terminal domain, let's leave the loop residues intact. Let's mutate the contact region instead. So we did that. And I'm only showing part of the data here, but as you make progressive mutations, and by the time you make all the 3 mutations, complete activity in the glutaminase site as well as the Afghan synthetase active site is lost. That means indeed, whatever we envisioned that the loop was opening and a dynamic interface was formed, that interface most likely has some weight, because otherwise if we had disrupted this interface, we would not lose the activity. So you can disrupt the interface from this side or this side, you lose complete glutamine activity by the time 3 mutations are made. Okay, so that is good. At least we got to know that there is the signal is being transferred via this active site. And here I think I may have skipped a slide. Maybe I made it more abridged, but let me tell the story from my side. What we see here is, which I am not showing you in our slides, is that there is a lot of rearrangement which happens in this region also. If you remember, I had shown the movie where this rearrangement, this region rearranges. You can see this loop moves, and so this region actually releases something called an oxyanion loop, which then activates the glutaminase domain. And also this changes here also open and close a passage, which we think maybe where glutamine is actually entering and leaving from. And if conformational changes happen in this region, you can maybe have glutamine access stopped if properly the whole system is not able to move and open and close. So there may be there may be a regulation which goes via this region up till here, and entry of glutamine could be actually stopped at this point, and also the activity of the glutaminase domain can be regulated via this motion. So at that point, at this point, we still haven't talked about where the channel is. We are still more interested in being able to pass the signal. So when first the structure was solved, there were 2 channels that were reported, one which was more direct and one which was a little bit indirect, which was passing via that ADP binding site that I talked about. We still don't know the role, but it caps the channel on one side. During the simulation, what we see is that this channel which was predicted turns out to be the channel that the simulations show. So here you can see the way what we see in our simulation is that the whole enzyme, it actually opens up during the simulation, and there is a passage which is formed where ammonia can pass through, and it is exactly the passage where there were some blocks that were there before when we were going to the structure. Those are removed, and you can see that these beta strand moves back, this helix moves a little bit, just enough to create space for ammonia to pass by. And there are 2 gates that come in the picture. So the channel is mostly hydrophobic in nature, and here there is a mouth gate and an end gate which is there. So this histidine is the end gate, and this particular region is the mouth gate, and you can see some kind of motion that is being seen in this region, and that is akin to the mouth gate. Okay, so what we thought was, let's look at this a little bit more in detail and see if we can understand the regulation of these gates a little bit. So we have a gate over here, which is an aperture type gate, and we have another swinging door type of gate, which is at the bottom. So we made a series of mutations. Actually, what we did was we made, I've abridged the whole story here. But what we did was we made several mutations. Some of them I'm going to talk about. Some of them actually I'm going to leave for this talk. But basically, we wanted to understand how does the mouth gate function. And you can see this mouth gate has a histidine, and it's hydrogen bonded to this serine, and also hydrogen bonded to you can say a salt-rich type of interaction with this D657 residue. And there's another residue which is N1051, which is very close to what I told you before, inoxyanion hole, which helps in activation of the glutamine hydrolysis. It stabilizes the anionic transition state, and it also talks to the downstream channel. You can see it's very close to the channel. So both D657 and N1051 are lining the channel, and these are kind of like the push that is required to get into the channel. So when we mutated residues, like for example, we got rid of this arginine and we made it an alanine. And what we saw to our surprise was that there was an enhancement in the activity. And even serine, when we kind of change to another amino acid, what we see is that when this hydrogen bonding is broken between them, then it turns out that the ammonia production increases. Okay, and not only that, we also see at the same time the FCAM synthetase production is also more. So the enzyme is slightly more efficient when you do these mutations. Okay, so that was a surprise for us because we were thinking most of the mutations are going to just make it dead. But recently, we also made arginine to many other, like lysine. We see that there's no effect on either activities, which I'm not showing here. And we also made it into a negatively charged residue, and negatively charged residue really hampers. It does not affect ammonia production, but downstream FCAM synthesis production is gone. But so you see that this mouth gate seems to have variable effect. You can also see that if you get rid of this D657 or N1051, in both cases, FGAM synthesis production is down. Okay, so ultimately, what I am trying to tell you is that the mouth gate has these subtle effects where sometimes the activity goes up, while at other points, when you're actually inside the tunnel, if you kind of get rid of the interactions, the tunnel opening and closing is affected, and the activity goes down. But if you go to the bottom of the tunnel, you know, if you remember this tunnel, this is the bottom side. There are amino acids which help histidine anchor and around it. You change anything in the bottom, okay, and 100% activity is gone. So what it looks like is the bottom gate is very sensitive. 100% activity means 25 angstroms away, ammonia activity is also gone. So everything seems to shut down if bottom gate is perturbed, but in the top gate there is a lot of variation, and the controls are much more tunable, I would say. So, looking at this, we also were able to get certain crystal structures of various states. And what we figured out through our work was that, like I told you, this D657 and N1051 are the key residues which transmit the signal to the channel for its proper opening. And if you mutate this, like N1015A to an A, this particular connection is lost. Or if you get rid of D657, again, the connection to the gate is lost. And as a result, both these two mutations are very detrimental. Whereas if you make any mutations where you break this hydrogen bonding, the flow of ammonia becomes much more easier, because these both of them are more free to move, and you actually get enhanced ammonia into the channel. So while we were noticing all this, we noticed another very interesting thing, which is a fact that we just pointed out in our work, and it needs more, I guess, deliberation. And that is that we saw that the ammonia channel, it is not like the ammonia channel opens and then it closes. What we saw was the ammonia channel opens during the simulation, and then it vacillates between open and close, and then finally it shuts. So there is something called a pulsation which is going on, where where it is not like once it is open forever. It is more like pulsating tunnel, where there are regions which there's a bottleneck over here, which kind of the residues go in and out, such that the ammonia channel is not 100% open all the time. So there is, we thought about it a lot. It could have many reasons, because there is more than one ammonia produced per cycle of catalysis in the second active site, and it could be a way to make sure that ammonia actually flows properly down. No solvent is there, and also maybe like a push to get ammonia into the second active site. So there are some things that we are debating, something that we don't understand. Maybe we need to do more experimentation, but I thought it was a very interesting thing that we kind of saw a pulsation in the tunnel that is there. So coming to the end gate, finally, I told you it is very, very sensitive, the end gate. Any mutations, the residues are among 1,000 purelles completely almost conserved. You tweak anything in the end gate and it's like bam, gone. So, but it is noticed that anything which is even in the periphery of the end gate, and this is Q471 that I'm showing you over here, this residue is on the C loop. And what we saw was that if you make this intermediate, it looks like the gate talks to the C loop via this amino acid. And during the process of the reaction, this histidine most likely donates a proton so that you actually get the reaction going. And at this point, Q471, you remember the loop is closed and it starts to open up. This is the residue which also moves as the loop starts opening up, and this connection is lost. And this is one of the ways the progress of the reaction via this hydrogen bond is communicated. And if you get rid of this residue, you see there is a 40%, more than 50% loss in activity. And our simulations results show that when you try to mutate this residue and you do a simulation, 50% of the time the signal passes properly, 50% of the time the gate is opened, closed, and 50% of the time it remains closed. And also the glutaminase domain, in 50% of the time it is not well oriented, and 50% of the time it gets well oriented for action, which tells you that there is a misregulation in the whole system that is introduced when Q471, which actually talks to and reports the progress of the reaction, is not being able to tell its story. Okay, so the regulation is multi-layered here, where you have the first. So what I would like to highlight here is that the progress of the reaction is directly read by the hydrogen bonding interaction of Q471, and as the loop moves, then the interface residues come into picture, and then you have the signal passed. So in conclusion, what I would like to tell is that this enzyme has multiple levels of controls. In the beginning, in its free state, this enzyme has no substrate in both the active site, but as soon as the substrate comes in this active site, this loop gets ordered, and there is this connection that is formed. And when the reaction happens, the first cycle of ammonia is always produced. When the reaction happens, this loop goes, talks to the N-terminal domain, and via the interaction with the N-terminal domain, this particular domain then crosstalk happens, ammonia tunnel opens, and flow of ammonia happens. And this cycle then keeps continuing. And that's how the enzyme is able to talk. So there is signals start at the second active site, and they travel all the way to this active site so that ammonia tunnel can open. So it's a allosterically regulated system where only upon entry of the substrate does the tunnel form. Right. So with this, I will end this part of my talk, and the main message that I wanted to pass was that how this complex interplay actually works. Now, the second part of my talk is actually much shorter, and it is just I'm going to introduce the system to you. So not a lot of time will pass, but I would like to tell you that this other system is also very interesting. And this system, we in the beginning only did a lot of work. It's a phenol sensor. We did most of the work which was in trying to develop a sensor, and we were more focused on trying to develop sensors for water pollution and monitoring, convert the sensor using the structure of the sensor domain to make it a benzene sensor, toluene sensor, and stuff like that, immobilize the sensor, try to multiplex the sensor and make it low ppb and stuff like that. But a very interesting thing that is going inside this structure itself is a lot of allostery that was going on, so that communication of the sensor domain with the adjacent domain, which is the readout domain, and stuff like that can happen. So here today, I'm going to give you our work that we are proceeding towards understanding the basic allostery and the function of this enzyme, not from a sensor perspective, but from understanding molecular mechanism. So this enzyme is a multi-domain enzyme. It has a signal reception domain where it actually senses this substrate called phenol. And then it tandemly located via this something called a B linker is a ATPase domain. That means it hydrolyzes ATP. And also it has a DNA binding domain. Now what this enzyme does is, this enzyme is a sensor of phenol. So when in the environment, phenol becomes very high in the bacteria which have this protein, they start using phenol as a energy source. because and then they shift their pathways to degradation of phenol. And this enzyme is like a natural sensor which decides whether to activate the pathway or not. And this is done in presence, whenever you want to activate pathways, you need RNA polymerase, and stress-related activation is done by this something called sigma 54 RNA polymerase, and it needs to be put into an activated state, and it is done by this enzyme. So it binds the DNA and that upstream of where the RNA polymerase binds, and via a mechanism where there is bending of the DNA, this comes close. And people also think that they form hexamer or heptamer units. They are mechanoenzymes, and they actually activate it. So the question that we asked was, how do these two domains, this is a much more complicated whole pathway, but how does this particular enzyme, only in the presence of phenol, results in activation of ATPase activity? So when there is no phenol, nothing, no ATP hydrolysis. When there is phenol, some conformational change happens, so ATP activity can happen. So we wanted to understand this in a much more detailed way. And so we wanted to understand the allostery in the system. Today, I'm not actually going to cover the whole allostery of the entire system, but I'm going to concentrate only on the A domain. We were able to solve the structure of the A domain, which is called, this enzyme is called MOPAR. That's a gene name. Along with this, we were also able to crystallize the B linker. So this linker which changes conformation so that the ATPase domain knows that phenol is there. So we wanted to understand this mechanism. Not many structures are there of the entire setup. Actually, no motor structure where the whole motor is assembled has been solved till date. So there's a lot of curiosity in the system as to how these mechanoenzymes work. And so the question that first we asked was, towards this curiosity, we made the question smaller. We tried to understand, because we have the structure of this A domain, we try to understand questions as to how does phenol enter. Two questions we had. And it was noticed that we tried to crystallize the enzyme in the apo and the phenol bound form. Every time we only got the structure of the apo form, of the bound form, sorry. Never could we find anything where the apo form was there. And the phenol is very buried inside. So it has to open and close, or something has to happen for phenol to enter. Otherwise, it will not be able to enter. So we wanted to understand, is the phenol bound form differently structured than the form of the enzyme, which is, APO? And how does phenol binding actually transmit the information across? Now, is it via only through the B linker? Remember, I said this B linker rearranges. How does phenol binding has anything to do with B linker? And are there any other regions in this protein which talk, or this is the only two regions which need to talk? So we did a simulation, multiple simulations actually, and we saw that not much conformational change was happening when you simulated the apo form of the enzyme. But there were distinct regions here, like we name it later the G hinge region, and like the B linker region that I was saying, they do show quite a lot of change. So you can see here, pocket region, which is where the pocket is, this region, this particular region where there is a G glycine residue, so we named it G hinge, and this region had some changes that were happening across the trajectory. Now, this also looked very similar to a G hinge region, which is found in many proteins when you want to do motions. Now, what would be the significance of this? So if you go back and look here, there could be that the enzyme needs to open and close, and it is via connection via here that it opens and closes so that the phenol can enter. Okay. So what we did was we looked at the APO and the bound form, and we looked at the networks that we found, and we found that there was a if you look at the crystal structure and the APO form of the enzyme, there is a phenol entry introduces a lot of rearrangement of the network. So you can see this network, there's a lot of hydrogen bonding that it is all gone when phenol comes, and a restructured hydrogen bonding pattern happens. And it also results in this hinge region, this helices moving much more apart, and this hinge region kind of moving. So So if you look here, I'm talking about this region and the network that goes across. We also saw that the network which goes from the phenol pocket to the B linker, that also completely, there is a shift in the network in the apo versus bound form. Once phenol enters, there is a restructuring of the network and this linker actually is released. And there's a conformational change in the linker that happens. Okay, but there is a lot of you can see that the hydrogen bonding pattern seems to be different. So what we decided was to experimentally pursue portions of what we had thought about. One thing we did was we said this hinge region may have some influence on phenol binding. Let's mutate the hinge to a proline, a more rigid residue, and see what happens. To our surprise, the affinity to phenol became 7-fold higher. This is experimentally we observed. And when we saw the structure, although we didn't see many major change in this region, we saw this whole linker had completely rearranged, rather melted, and a new helix was formed. So things seem to be traveling to the mutation made here, changed affinity here, and also changed things in this B linker. So looked like the protein had more connections that we initially thought about. Okay. Now, we did more experiments, which I am not going in today because otherwise time is of essence. We did a lot of fluorescence experiment to show how various tryptophans strategically located here will change the, the conformation apo and the bound form. And we found several changes that were subtle but important for function. But one thing that came out was that there was a allosteric network which seemed to be connecting these regions. So this region and this region through the electrostatic network that I showed you and this region are actually connected. So it could be via the pocket that the connection is there between these domains. So we decided, why don't we perturb the network a bit? So we chose some amino acids and we in this network and some in this network. The red ones are shown here, and we also found in species which have bind benzene and other enzymes which form similar molecules to phenol that have not been structurally characterized. They also have similar networks. I mean, these residues are conserved in those. And anytime we try to break the network, what we see is that immediately the activity, which is the ATPase activity, which is the hydrolysis activity. So unfortunately, I should have put a picture here, but basically what I mean here is this activity of the C. So we made a construct which had these domains, and we see every time we made mutations in this network, phenol entry network, the relay network, we would see almost no ATP hydrolysis stimulation that is happening. So indeed, these networks have some role. This is ongoing work that we are doing. So we kind of feel that internal hydrogen bonding and some electrostatic networks, they are at the core of the protein, and you may not see a lot of conformational changes within the domain, but these subtle hydrogen bonding network changes may actually contribute a lot to function. The second question we asked was, how does phenol actually enter? So here, let me show you. If it would let me go back to my laser point. Yeah. So here we can see this is phenol, and this is just a short movie which shows how phenol actually finds. How does phenol find its ligand? Okay, this is what we are trying to understand. You see, it went in. There was no major conformational change that it opened up, because if there's a major conformational change, many molecules can enter. But you saw that phenol ultimately in the simulation found its path. There were I'm only showing one of the paths, but most of the paths, there was only one predominant path that we found in the simulation. Again and again, that would come by which this entry was happening. There was another path which was very small number of times we saw. But when we looked at this path a little bit more carefully, what we see is there is stages in which phenol enters. Stage 1 is at the lip, and then stage 2 is it goes in the middle, and then stage 3 is when it is actually in the pocket. So stage 1 is it's here that it enters more, and then it enters more. So let me show you in a zoomed view. This is the lip where it enters, and phenol is trying to enter. Then it enters. That is stage 2, and then it's completely entered, which is the stage 3, which is inside the pocket. So what we did was, we saw this pathway by actually simulation. We wanted to prove if this is real or not. So we made a lot of mutations. We actually made it a little bigger, the hole over here, so that phenol can go easily, so without changing hydrophobicity. We blocked this pathway by making 2 types of mutations: mild polar mutations to salt bridge. Okay, and we also made some at this region where F19 and F132. We're still making some mutations here. So let me show you our results here. What we did was, I don't. There are more than a couple of mutations we did, but let me show you one example where we made this methionine to a glutamine and this to an asparagine, which is amide. Both of them are amide. Some kind of hydrogen bonding can happen, and you can see that 10 times reduced affinity of phenol happens when we do this. And not only that, if we make it a salt bridge, there is no affinity at all. Phenol cannot enter anymore. You just completely lose. So you make it less conducive here, but here you completely kill it. Okay. So indeed, if you are trying to seal this lip, let's put it that way, then and you make it too hydrophilic, then phenol is unable to enter this lip properly. And similarly, we did a similar mutation in this. Once it enters the second stage, this is where in the simulation we see that it goes and sits, and there is this F19 and F132. So far we have made conservative mutations where we want to see how important the side chain is. And we see that out of the two, F132 seems to be much more important for you lose the affinity much more, about more than 10 times when you actually mutate this residue than this residue. So what we are trying to prove here is indeed what we see in the simulation: stage 1, stage 2, and stage 3. Stage 3, we have a lot of data already when it is already in the bound in the pocket. But basically, this is the pathway by which phenol enters. And so how does phenol know how to enter, and what are the ways by which phenol actually finds its pocket is the answer that we were trying to show both computationally and experimentally. So with this, I just want to end my talk. And here we have dealt with this system where we see that mutations which are far away, they are 20 angstroms away from the phenol pocket, have a profound effect. And it could be that what we think is a domain where only one phenol pocket is there and nothing else is talking is not the case. There are many networks which flow inside the protein, and they are actually have their significance, and this we feel is helping this pocket open and close, and is involved in that part. And it influences both the G hinge and the allosteric region influences both the downstream activity. We have done, I did not present here, but we have also done G hinge activity studies, and we see that not only does it bind phenol better, it also has a higher transcriptional activity. So it could be it is actually a more efficient mutation, which has resulted in the phenol actually being more conducively, and the network is not getting spoiled by this mutation. But if we take mutations where we spoil the network, we do lose activity. And the second question is, does it really enter through this pocket lip region that we were initially thinking? And we believe that yes, it does enter through this region. So these are two questions that we have been probing with this, and I thought that we could touch upon both of them. With that, I end my talk, and I want to thank all the people involved for the first part of the work. Nandini is the prime person who did most of the work. Even the simulation, most of it was analyzed by Nandini, although Navjeet and Padmani helped a lot. Navjeet with the expertise, he ran the simulations, and Jagannath has been our major collaborator in this particular work. I don't have, unfortunately, Sahil's picture, but Sahil and the second part that I talked about, Jayanti, are working on the system, and I try to abridge it in the interest of time. I think I've already taken quite a lot of time. So with that, I would like to thank everyone, and I'll be welcoming any questions if there are any, and thank the funding agencies also. So thank you.
Thanks, Ruchi. It was really fascinating talk, and the floor is now open for questions. If anybody has a question, Ubinandan has posed a question. Shall I read it, or you'll be able to?
Just 1 minute. Yeah, you can read it. Let me try to get back to the
Okay, I'll read it. It's a long question, so that's why.
Yeah, is it a very long
Okay.
question? Should I read it? It's in the
I
chat.
agree. The end gate is for the release of products. Are the end gate residues more conserved? Did you try to mutate the bottleneck residues? Can you just brief how did you trace the allosteric pathway? I mean, how do the allosteric site communicate with the active site?
I guess. It's a very complicated. I mean, first time, and if I were the first time listening to it, it is a little bit more complicated. So there are two active sites, and this is the end gate. End gate is like a door which opens and closes, and what we see is once the end gate opens, it opens for the entire time of the reaction, and what the door which is opening is actually also participating in the reaction. So yes, end gate is very conserved, and any amino acid which is near the end gate is also very conserved, not which are not a part of the active site, although end gate is very near the active site. And what we saw was any mutations we made in the end gate resulted in complete loss of activity at the other ammonia binding site, which is 30 angstroms away. So end gate is very, very important. And we think that when the substrate FGAR binds, the end gate actually opens up because it has to participate in the reaction. And that's how it remains open throughout the reaction. And any perturbation of and this results in a loop because the substrate is also talking to a loop, and that loop then starts opening up, and when it's partially open, it talks to there is a domain in the middle. It talks to it, and by which the information passes back to the ammonia production domain. So it is like a network in which once the substrate enters, the end gate opens, it passes the information, and the other domain gets it, and the ammonia channel is open. That's how it happens. I don't know whether I answered the question.
Did you try to mutate the bottleneck residues?
Actually, I not exactly, but I will. I have to go back here because I have a data which I saw, which I will. I did not present. I actually abridged the portions of it, but I think I have that slide there. If you look here, you can see perhaps. So if I go back to my talk and I show you the bottleneck. There are a couple of bottleneck residues. So if you see here, we actually, the main bottleneck for the pulsation is, the main bottleneck for the pulsation is around this region, but then there are the residues which have to move, lie here and here. So what we have done is, we had the whole tunnel in two states. So what we did was, if we try to do very drastic mutations, what will happen is the protein will become insoluble. So instead, what we did was, you see here this valine. This actually moves away during the course of when the tunnel opens. So we converted that to something called an isoleucine, which is slightly has one more carbon, right? So you can see here, and then the tunnel most likely we thought will now close by few angstroms, or here you can see by 2 angstroms, and we actually were able to get the crystal structure of this enzyme with this tunnel slightly closed. And we see that this enzyme is 50% less efficient. So what we see is ammonia production, nothing happens, but ammonia, there's a lot of leakage that happens in this enzyme, which is shown by how much ammonia leakage, and also FGAR production is 50% gone down. So that's what happens when you try to actually take whatever channel you have and you try to kind of block it slightly. I don't know if I answered the question fully, but we experimentally tried to block the channel a bit. But exact pulsating residues, we have not yet mutated many of them.
Any other question? You can raise your hand. Yeah, Manoj, please go ahead.
Okay, so yeah, nice talk. So you have strong mutation effect in your ammonia channel. So
Say that again.
So like you observe very strong mutation effect, like in your
Yes, yes. In some cases, yes, yeah.
yeah. So did you observe any like pH effect? So I want to say, so I want to know because like whether it is ammonia or ammonium, or there could be a single ammonia or multiple ammonia.
Okay. I'll try to answer your question a little bit. I don't know which it will be accurate or not, but one thing we have done is that in this enzyme, if you just give no ammonia, but you give ammonium ions, you put a lot of ammonium ions, then what happens is there's no reaction. This enzyme, you can flood the whole system with ammonium. You can add different kind of ammonium salts at different pHs, but it does not respond to ammonium at all. Okay, so that is one thing that we really know about this enzyme. When we try to look at the catalytic rates, we believe that there is always some leakage of ammonia, and more than ammonias are produced, or 2.53, something like that, per catalytic cycle. So for every cycle, we think that the ammonia production is slightly more than that of the F-gas synthesis. And ammonia reaction is actually way more also efficient. If you would to leave this enzyme without substrate, we actually see that the rates of ammonia production are very high, much more higher. Now, another thing that we have noticed is if you go slightly higher in pH, this enzyme becomes completely dead. Above 7.5, if you start going, then enzyme becomes completely dead. Now that there, it's a different effect. Ulta hai. We have never tried to see. I actually forget. There is a bell-shaped curve, but what pH it loses its activity, I don't remember. But ammonia presence we feel is very important, because without the nucleophilic ammonia, you cannot have the reaction happen.
Okay.
So partially, I think I understand.
Anybody else? Okay, maybe meanwhile I'll ask. I have several questions. So first thing was, do you have any estimate of the time scale of the communication between these two sites? That once the substrate binds, how long it takes for it to propagate? Do you have any estimate?
We only have simulation timescales right now.
Yeah, that's fine. Either from experiment or simulation, that's fine.
So we think it's a millisecond event right now, but we are not sure. But we are doing now experiments. What we are doing now is we are doing coupled kinetics. We had some problem with having FGAR, the substrate. It is a very difficult substrate to make, and we are also tagging the enzyme with unnatural amino acids with fluorophores to confirm that. But I'm not sure yet. I've given you this answer. That's what we think. But we are actually pursuing exactly what you've asked as a future experiment.
So I think, I'm not an expert of the experimental techniques, but it's possible to freeze the intermediate conformations using this cryo kind of techniques or not? It's not.
This enzyme, once it starts going, we could do a FGAR addition and see if we can find. Yes, kind of, we can freeze because we have slow catalyzing. So exactly, we have slow catalyzing mutants now, you saw, right? So we could try to freeze some of it. We don't know what we're going to get. We may get an ensemble, and one of them may be the correct one. And it's 140 kilodalton, and we are now at the place where cryo may be able to do it, because crystallography we failed a lot, but we are also, like I said, trying to understand dynamics by having unnatural amino acids where we have fluorescence tagged, and we have actually already developed that system in our lab where we can move the fluorophore throughout the enzyme and be able to map. We're ultimately trying via FRED to map the conformational changes that we have observed. Right. I don't know, one day maybe it will be possible. So we're still designing those experiments.
Yeah.
And we also do coupled kinetics now. And the reason we were not able to do it is, for a long time we had in our lab, we were not able to make a lot of, we used to get our substrate from Johann Stubbe's lab at MIT, but now we are making our substrate ourselves. So we can now have a large amount of substrate that we can do many coupling kinetics.
So that pulsating movement that I found really interesting. It's similar to kind of this peristalsis kind of movement.
Yes, we found it very interesting, but I think we'll have to prove it experimentally.
Yeah. So I was wondering that actually this is again from the simulation perspective probably, that so it should be something like when it binds at a particular site, it should open up in the front. So there has to be some kind of a correlated fluctuation that there has. When it why it goes on. I mean, I was trying to think that why it will go in the front direction, because when it binds, the next step has to go forward.
That's so I
opening in the front.
thought maybe
when
it will
goes,
pulsate
it has
like
to go
this.
back. Yeah.
You know, so we were trying to understand this. It's actually very complicated,
Right.
and we are really looking. You don't know, we never even noticed. And to be very frank, we published a science advance and we did not notice the pulsation, because it was like we were so hell bent on just looking at the channel, and we were so awed by getting the channel that we just got like into it. And then with time we realized, we kept on looking at the snapshots, and then suddenly we would not find the channel, then we would find the channel, and then it is now that we are able to find the pulsation, and it looks like it's pretty. I mean, I don't know, but we think it's periodic. We saw it a couple of times. It's not like just once. But is it like we have captured it properly? We are still not. First we thought that we should just tell the world it's pulsation, but now we have toned it down because we don't have enough experimental evidence to prove it. But we thought we should get the thought out there.
Yeah, because I have been curious that so when you have a charged molecule, there can be electrostatic guiding for the driving. So, but for a non-polar gas molecule like even oxygen, so how is it driven?
We have actually read a lot of papers in this now, and we see that there are many enzymes which they do not call it pulsation as such, but they call it some kind of breathing and stuff like that. And it is pretty common in gaseous enzymes that they go through some kind of, but so it is not.
This could be more general. This could be general for any, I mean,
yeah,
like
so
active diffusion of gas like.
Yes. So we looked up, actually recently, we looked up this oxygen tunnels, carbon monoxide tunnels. We have looked at so many gaseous tunnels to see, for gaseous molecules, it becomes more important to have pulsations because they have no other way. There's no contact with anything else. So they can always reflux, right? Back. So there has to be a mechanism by which reflux is prevented. So we believe that many of these systems actually have pulsations, it's just not recorded.
Right. And my last question is again kind of general, that here also you find that this hydrogen bonded network seems to be very important. Now, if you try something like, let's say, techniques like SCA, for example, statistical coupling analysis, we have tried this earlier, but it seems that SCA always gives you non-polar residues. These pathways never match. So I was kind of curious that first one, that how general is that it's always this hydrogen bonded network, that electrostatics driving the allostery, or there could be some other kind of interactions like van der Waals or hydrophobic can be important in some cases. And what I find that when you use methods like SCA, you never get this kind of electrostatic network.
On the Purell system, we have done SCA, and we had a very major problem. We were never able to actually, we did find, like you were saying, we found a hydrophobic patch, which was mostly, we were only finding, we were trying to understand where xenon goes and binds, and many times we were not looking for that. We were looking for the tunnel actually. But every time with SCA, we were able to find the most hydrophobic regions.
So you have the same experience also.
So then I realized, but we were not able to get the allosteric pathway by that method. And anything. So we actually gave up on the SCA, to be very frank. So we, couple of years ago, we did try SCA quite actively, thinking that we are just going to find the allosteric network. Okay. Then now I have, to be very frank, less confidence in that. And here we did a lot of, we've done hydrogen bonding propensities, electrostatic interaction, to be put it generally. And we also did hydrophobic interaction. We didn't see much change. Actually, I have not, maybe we should put it in our paper to show that we didn't see much change. Right. But because I kept on telling them that, no, find the hydrogen bonding, find the hydrophobic interactions also, maybe we're missing. But every time Sahil and Jagannath and I had a meeting, we did not see much. That's the truth of the matter, actually. But we saw this electrostatic interactions really and change. And what we have seen is that if we go across members which are like phenol, like I told you, we also saw that we don't have many members that people know it for sure, about 4 or 5 members. All of them have these residues. Right. And the ones where the main chain is involved, they are completely conserved. But the ones which are side chain, there is variation that happens. Yeah. So that made us believe that the network was real. Okay, that's why when we started doing mutations in the network, and what we saw was, which I also did not, we're still working on this paper, What we saw was even when the phenol binding was happening, relay was not happening. So what happened was you were able to bind phenol, but no ATPase stimulation was happening here. So those mutants that I was showing completely zero activity, many of them can bind phenol, but they're not able to transmit that they have bound phenol to the next domain. So it becomes a more of a problem that the binding information is not going.
Yeah.
So
I think it intuitively makes sense that electrostatic interaction on hydrogen bond gives a more directional nature, because these are like, there is a certain orientation that it provides a direction to a network.
yeah,
hydrophobic interaction is more isotropic. Yeah. Right.
So I actually, we actually observed this after we did the simulation, and there was not much conformational change which changed the whole, it was not like the whole system just became something new.
Sure.
So we kind of believe that earlier I was thinking phenol will come, huge opening. Then I realized that if phenol opens so much, any molecule can go in.
Sure.
Yeah, right? Exactly. Then how the selectivity will be maintained? So obviously it has to squeeze in through its a very, orchestrated motion.
Sure. Yeah, it's very interesting. Yeah. So maybe we'll take the last question. There is a question from Shomadip Sarkar in the chat box. Why did it increase in one of the mutations in the production of glutamate?
So there is this mouth gate where there's an arginine residue and a serine residue, and they both hydrogen bond. Okay, and ammonia is supposed to come from there. Okay, this is the active site, and right outside the active site they are. Still the hydrogen bond is maintained. There is some control in which way the ammonia will be produced. But when you mutate these residues, they kind of move. Either it's not there, or in one case it moves in the second way. In the hydrogen, this bonding is lost. So here now what happens is also arginine is slightly positively charged, and ammonia is being produced. Here now what happens is the control of ammonia production is completely. Is not completely, but slightly compromised. So it can fall into the system much more easily. And if you change the flow slightly, not a lot, then it is able to, the FCAM synthesis active site is able to, kind of process the whole thing faster. So it's a slight enhancement in the overall activity, because the controls, the rate of ammonia flow has slightly been altered because this hydrogen bond, which was kind of controlling release of ammonia, is gone. That's why the serine and the arginine mutations are the only mutations which increase activity.
Okay, I think if there are no further questions, let us thank Professor Ruchi Anand once again for a beautiful talk. Really lots of fascinating results and insightful discussions. And Ruchi, you have an open invitation that you should visit our campus sometime when under better circumstances things become better.
Definitely.
So thank you again.
Thank you so much, and I enjoyed discussing my science with you. Thank you all.
Sure.
Thanks a lot.
Sure.
Good
okay.
evening.
Thank you, everybody. Take care and stay safe. Okay.
Bye.
Bye. Bye.